All right, thank you, Carol. Thank you. Well, thank you everybody for being here. It looks like there's 123 participants. So thanks everybody for coming to listen to my colleagues, Katie and Jen, and I get the honor of introducing them. I'll start with Katie. Katie, wave to everybody. Katie, there she is. All right. Katie Pearson, she completed her master's degree at the University of Florida and studying shifts in flowering time in members of the sunflower family. And so she's a real phenology expert. And during that time, she worked a lot in the herbarium. She's going to be talking a little bit about that tonight. She was active in digitizing efforts and became an expert in digitizing herbarium specimens, and then came to Cal Poly 2 years ago to manage the project which Jen is going to talk about tonight. Currently, she actively coordinates all 22 herbaria in California on the project that they'll be discussing tonight, and trains countless other herbaria on digitization and photographic efforts and so on. She has been, and currently I'm teaching a class that Katie is a TA for, a class called Plant Diversity and Ecology, and she has been a wonderful addition to Cal Poly for our students. And unfortunately, her first teaching event has been online, but we will come back to in person soon, and Katie will be a star teacher, I'm sure, in person as well. And she's been a great addition to the Hoover Herbarium here at Cal Poly as well. So Katie, thanks for being here and welcome. You'll hear more from Katie towards the end of Jen's presentation. And there's Jenn Yost. Jenn Yost, wave hi to everybody. Dr. Yost was she actually, she has her mask on. That's good. You're alone, so you don't need to have the mask on. Jen was an undergrad here at Cal Poly and got what very few people know. She got a master's degree in marine biology here at Cal Poly, and then something switched in her, and she became a plant biologist. She went to graduate school, got a PhD at UC Santa Cruz, studying the evolution of flowers in California, particularly studying the genus Lysthenia, and that's goldfields for all of you who don't know that genus. And then we hired Jen at Cal Poly in the biology department in 2014 as a ecology professor and as the director of the Hoover Herbarium. And she has taken the Hoover Herbarium up 200 notches compared to where it was when she arrived. She really has done some amazing things and has become a pillar of the California botanical community with regards to the herbaria, and she's leading this massive project with all these herbaria that she'll talk about tonight. It's Jen is also an incredible teacher. I've had the honor of being able to teach with her. We teach a field botany class together, and I get to watch her really fire students up on the California Flora, and you'll get some of that tonight, I'm sure. So Katie and Jenn Yost, welcome, and I'll turn it over to you too. And I'm going to be monitoring the question and answers, and so if you guys have questions, go ahead and put them in the question and answer section or the chat, and I'll look at those, and then I will moderate that at the end. I'll ask you guys some questions that the participants have. So thanks for being here.
Thanks so much, Matt. All right, I hope that everybody can see my screen. Can you see that okay, Katie and Matt? Yep. Great. For those of you who are watching the webinar, it's Jenn Yost and Katie Pearson. Thank you, Michael, and thank you, North Coast Chapter, for having us and putting on this webinar series. It's sort of like the sister connection between the San Luis Obispo chapter and the North Coast Chapter. As Matt said, I am normally out in the field during the spring teaching young people about plants, and we've been faced with so much bad news lately that I wanted to plug the fact that botany is alive and well at Cal Poly. So Matt and I are just a few of the incredible botanical faculty that are based at Cal Poly in San Luis Obispo. We have Dr. Nishi Rajakaruna, who studies serpentine plants, Matt, of course, who studies all aspects of trees in California, Dr. Dina Grossenbacher studies the evolution of the California flora. Myself, and then the godfather to all of us is Dr. Dave Keel, a plant taxonomist and a researcher of the California flora. And all of us have the incredible opportunity to take young people out into nature, and we all teach many, many field courses. And it seems like in a time when many institutions have cut their field programs, Cal Poly's has persisted, and it's a very wonderful thing. And it's great news for people who care about native plants, because I feel like we're this pipeline bringing young people to botany all the time. So tonight, what I want to talk about first is a national effort that has been underway that you might not have heard about. So in 2010, the National Science Foundation decided that having our nation's museum collections digital and accessible online was a national priority. And so they funded a national center. That center is called iDigBio. And the goal of that center was to help all the museum and biological collections of the country digitize those specimens. There were estimates when this program started that there were between 500 million and 1 billion biological and paleobiological specimens in the United States. And those have been collected over the last 200 to 300 years. The problem with most of those specimens is that they were sitting on shelves or in the basements of museums, and nobody had access to the data that they contained. And so the goal of this effort was to bring those specimens to life. And so what this center did was coordinate effort among groups of people who would get together around some research theme and then ask for funding to digitize those museum specimens to address that research question. And so over the last 10 years now, here's a map showing all of these institutions who have received funding at their museum or herbarium to digitize records. And all of those digital records are then aggregated into this massive database. Currently, the iDigBio database has over 121 million records, and they span all aspects of life. So you'll find vertebrate collections, insects, fossils, and of course, many of those come from plant specimens that are housed in the nation's herbaria. So what is an herbarium? An herbarium is a room full of metal cabinets that sometimes smells funky, right? But inside those metal cabinets are a bunch of dead plants glued to archival paper. And these records are the only physical evidence we have of what plants occurred where. And so I'm going to make the argument that this is truly a profound exercise and a profound human endeavor. Specimens make their way into these cabinets first by collectors in the field. Collectors go out with their plant press and their notebook. They collect the plants, they take notes about them, and then they deliver those specimens to an institution, some herbarium. Once they're at the herbarium, they're mounted or glued onto this archival paper, and a curator turns those sloppy notes into a nice label, and then those specimens are incorporated into the collection. And now the push has been to take all of that information that's housed on those records and digitize it, make it searchable online by putting all that data into a computer. And so I'm going to talk a lot about digitization, and part of what we're doing is hoping that you'll help us with digitization. And so what that looks like is for every single specimen, they have a label. We need to type out what's on that label. Labels are written in every diverse way that you could possibly imagine, and we need humans to parse that data into the correct and standardized fields. And then we need to put a GPS coordinate on it. So we need to know where that specimen was collected in such a way that we could map it. And nowadays we're taking images, high resolution images or scans of each individual specimen. And so while that whole process seems simple, it truly is the foundation for what we know about plants. So the question is, how do we know anything about plants? Well, I would like to argue that whether you've realized it or not, it's because of an herbarium that we know anything about plants. So herbaria are the foundation of knowledge about plants. So I'm going to talk about their importance in understanding how we know what we have. And that's in regards to the current diversity we have in California, new diversity that we discover, and also the genetic diversity within populations and among species. How do we know where things are? And how do we know what's changing? And I'll talk briefly about range shifts, extinctions, invasive species tracking, and flowering time shifts. So what can we gain from an herbarium specimen? Well, they come with a label. Those labels have the name of the specimen that's glued to it, the authorship, the person who authored that plant name. They have a location, in this case, Black Lake. There's sometimes habitat information in wet ground at the edge of the lake. There's often associated species that were found growing with it at the time. There's the date, the collector, and most collectors assign a collector number. And that information is wonderful, but really the only two bits of information that are important are the date and location. Everything else can be reinvestigated later. If we have the date and the location, this specimen is valuable for research because the specimen tells us many, many things. So from looking at the specimen, you can learn almost everything you could learn from looking at a live plant. First, you can look at its morphology. Is it flowering? Is it fruiting? In many cases, colors are preserved on these specimens. You can determine the size of things. What's the length and width of the leaves or the petals or the sepals? You can look at the surface features. What are the trichomes like on the stem? What are the trichomes like on the abaxial surface of the leaf? In many cases, you can take these flowers which have been dried for maybe 100 years, and you can rehydrate those flowers and dissect them as you would a living flower. These specimens also contain seeds in some cases. People have looked at seed longevity using seeds extracted from herbarium specimens that could range from 5 years old to 200 years old. And then these specimens, each one of them, because it was an actual living plant, contains DNA that we can extract off the specimen and use for research. So how do we know what we have? Well, how do we know what's in California? You might remember from Matt's webinar that in California, we have over 5,000 native species and we have over 1,000 non-native species in the state. How do we know that? How do any of those authors who authored those gypsum annual treatments know what is in California? Well, they only know it from looking at herbarium specimens. And so you might ask yourself, is there more than one species of Clintonia in California? Matt and I were just up your way, and I got to see Clintonia andrewsiana for the first time. I was totally floored. It was a beautiful specimen. So I featured it in tonight's talk. So how do we know that there's more than one species? Well, if you were in the North Coast ranges or the Klamath, you might be looking at this multi-flowered, pink-flowered lily. But to know if it was the same or different as this other species, Clintonia uniflora, you would have to travel to the Sierras and hope that you catch that one and flower at the same time. And by the time you got there, you wouldn't remember what the one from the coast looked like. But the beauty of the herbarium is that you can go in, you can pull specimens off the shelf that span massive distances in geography. They can span time. I like to say that it's always spring in the herbarium. It gives you an opportunity to look at California's diversity side by side and try to make sense of it. And so these records are the foundation for how you ever see any count about what is in a flora and what isn't. All plant names, every single scientific name references an herbarium specimen. We call these the type specimens. The rules that are laid out for botanical nomenclature say that in order for a name to be validly published, you have to designate one herbarium specimen to be the reference for that new scientific name. And so this is the only way that we can make sense of what's what in regards to the use of scientific names. Here's an example of a type specimen from your area. This is Salix delnortensis from Del Norte County. This was collected by Alice Eastwood in May of 1907. And then a little over 10 years later, it was described as a new species of Salix. This is Alice's original label where she just gave it the genus Salix because she couldn't put a species name on it because it was undescribed at the time. The type population or the where this specimen was collected is Del Norte County on Waldo Crescent City Road in Gasket. And so this type specimen lives now at the California Academy of Science. And so if anyone ever collects another willow, let's say in Humboldt County, and wonders if it's the same as the one that Alice Eastwood described, they have some record, they have some specimen to go back and look at and compare. And if they unknowingly don't look at that and they name their collection a new plant name, we have a track record where we can go back and trace invalidly published names or correct names, and that happens all the time. Another important role for herbaria is that they are where undescribed species sit. This paper came out in 2010, which was estimating the number of new species that are sitting on the shelves of herbaria not yet described. And based on their analysis, they estimate that there are 35,000 undescribed species sitting on the shelves of herbaria today, waiting for an expert to look at them. And they did an interesting thing where they calculated the amount of time between when the specimen came into the herbarium versus the date at which it was described. And they can say it can take as long as 50 years for a specimen to be described. So this is a problem for many reasons. One, it speaks to the fact that we need taxonomic expertise and we need people investigating these records. If you want to describe a new species, you need to go to your local herbarium right away. I'll give you two examples of how herbarium specimens are used for describing new species. Many of you might have heard of this species, Adianthum shastense, the Shasta maidenhair fern. This is a great example of new species being discovered in the herbarium. This species was described in 2014, and at that time, the authors were studying Adianthum populations in California. As part of their work, they sequenced DNA from all of those 13 records, and they found that these Shasta collections didn't match the others. They didn't match what they had been identified as, and that gave them a clue that these might be different. And when they looked at their morphology, in fact, they found many morphological differences, so they were able to give it a new name. If you look at the collection history of this plant, it's fascinating. The first collection of this species was by Milo Baker and Frank Nutting in 1894. Then, almost 100 years later, Dean Taylor collected that specimen in 1992. And then it wasn't until 2014 that it got a new name. That's fantastic. And so everybody needs to be investigating the sheets on their shelves. When this new species was published, they had to designate a type specimen. And in this case, they designated an herbarium specimen that is housed at the University Herbarium at Berkeley. And this is that type specimen. So now if anyone has any questions, they can go back and look at this to determine if what they are looking at is in fact this new species or something else altogether. Another very recent discovery just published, a new name, is the Nelson string flower. This is Silene nelsonii. This is a species in which the authors saw this Silene and they referred to the type specimen for what it had been identified as. And this specimen did not match that type specimen. They were clearly different. And so based on that, the authors were able to make the case that this was in fact a different biological entity than what that type specimen reflected. Here on the map is showing you where in your area you can find the species. The little black dots are the new Silene. And when they published this new name distinguishing this new species, they designated a type specimen, and that type specimen lives at the Humboldt State University Vascular Plant Herbarium. So herbaria are important for many reasons. Another thing is that we can get DNA out of them. And I love that we can get DNA out of these specimens. I love it because when collectors, the vast majority of collectors, made these collections, they never imagined what studies would come out of that. And the value of these collections has just been increasing over time. So now we can extract DNA from even very old specimens to figure out the levels of genetic diversity in populations, even the level of diversity in populations back through time, how that diversity has then changed. We can detect evolutionary shifts or natural selection in certain populations. And most importantly, we can understand how species are related to one another. The image I'm showing you here is the monkey flower phylogeny, which was largely built out of extracting DNA from herbarium specimens. In addition to herbaria collections telling us what we have, they're crucial in us understanding where plants are. If somebody said to you, where do redwood trees occur? Of course, everyone in your chapter would be able to answer that question. And if you Google it, you will get this map. But the question is, how do we know if this map is accurate? Well, we don't know that unless we have a voucher specimen. I often say, well, did somebody drive around looking? Is this map generated from Google Street View? How do we know where the redwood is? And you might imagine that field notes would say something like, well, there were many conifers with needle-like leaves. Well, that doesn't help you at all, because that could be every conifer that you find in the north. Redwoods were very big at the canyon bottom. Okay, well, that one at least is referencing a species, but how do you know that the author could tell a redwood that you see on the left from a grand fir, which you see on the right? We don't know unless we have a physical specimen. And so everywhere that the California redwood has been collected allows botanists to make accurate maps of where those species occur. And that is the case for all species. What is the range of Ceanothus prostratus? Here's a map showing little pins for every collection there has been of that plant. And that allows us to understand anything about the biology of this species. In addition to knowing where things are, we use herbarium specimens to understand how things are changing. And this has become particularly important for California. It gives us a baseline. Here's a great example of how we're using herbarium specimens to understand how ranges might shift. So how will species move in the future? What I'm showing you here on this map, the light pink area is the present range of Camebatia foliolosa. And this dark pink area is where its projected range is projected to be in the future under climate change scenarios. And so we actually would expect more suitable climate for that species to be in the North Coast ranges and throughout the Klamath. That's interesting, but it's even more interesting that we see the opposite shift in something like Abies magnifica, where its present range is in the light green and it's actually projected to shift south in the future. This represents one example, but if we're going to care about shifts across the flora, we really need to know this for all species. It's given this kind of information that allows us to produce maps like this, like where in California are we going to see losses of endemic species? And what I'm showing you here is just 4 different climate change scenarios and where those species extirpations are going to occur. Herbarium specimens are also important for telling us what we have lost. And so I'll give you an example here from San Luis Obispo County. The plant is Arenaria paludicola. This is the marsh sandwort. The marsh sandwort, historically, if you look at the Jepson, the map looks like this, but historically it was known from only 3 populations, 2 of which now are extirpated, San Francisco and Santa Cruz. So the remaining populations are in San Luis Obispo County. Well, if we look back at the herbarium record, here's the collection from Black Lake of this plant by Robert Hoover in April of 1947. He went back later that same year and collected it. This is now a specimen from July of 1947. Well, Dave Keel followed Bob Hoover and was a faculty member here. He went to Black Lake and he found this specimen in 1985. But since 1985, it hasn't been found. Without these 3 specimens, we would not know that Arenaria ever occurred in Black Lake or that it had been extirpated from that site. And that's a huge deal when we start to think about rare species mitigation or rare species loss. There's so many specimens where I look up the label, I look up the data, and I try to go find it. And what's there? A strip mall. And that is very important because humans are bad with the shifting baseline, slow change over time, when all of a sudden we realize, whoa, we have lost an incredible amount. So that's an example of how it's important for understanding what was once here and might be here no longer. We also use these specimens for tracking invasive species. So here's an example currently from San Luis Obispo County, but watch out North Coast. This is Ehrharta calycina, perennial veldtgrass. It was first collected in California in 1940 down in San Clemente. By 1956, it had made it up to San Luis Obispo County here on the Nipomo Mesa. And by 1958, it was in the Santa Lucia Mountains. So what I'm showing you here is a map where every blue pin is a collection point of this specimen. And since we can map where the species occurs, we can also overlay things like temperature and precipitation, and we can make some guesses about what is the suitable habitat for this plant. And what that is mapped on this map in red. And so you can imagine that if you were in the Sacramento Valley or along the north coast of California, those areas are suitable habitat for this invasive species. And if it does anything to those areas, like it's doing in San Luis Obispo, that is a species of high concern for management. Many of you might know French broom from Northern California, another problematic weed. The first collection of French broom was from the year 1900 down in LA County. In your area in Humboldt County, it was first collected in 1906 by J.P. Tracy. And in his first collection there in 1906, he said, it is in the immediate vicinity of Eureka. But by 1941, when Tracy came back, it was now on the outskirts of Eureka. In order to understand these plants, how they're behaving and where they are going, we need to be able to map them. Now, I don't know how bad of a weed you all consider French broom in your counties in Northern California, but in the California database, we only have 37 records from your chapter's 4 counties, and only half of those have a GPS associated with them. Many of you might have actually helped to remove French broom from a local area, and the next time you do, you're all charged with making a collection of it and sending it to Humboldt or to Chico, because we need more records of where these plants are. And so when I went to look up the occurrences of French broom in your 4 counties, and when I realized that so few of them didn't have a GPS point associated with them, I thought to myself, this is a huge problem because people in Northern California know where these places are. And so that brings me to this short aside, which is that we need your help with georeferencing. So for example, I'm going to show you some localities of records that do not have a GPS coordinate on them, and I also have no idea where they are. So from Humboldt County, near H and S to Mill, east of Arcata. Xavier's Place, Bald Hills, Liscombe's Hills, Mud Spring Camp. Some of you might know where those places are, and we need your help to be able to put a pin on the map. From Trinity County, North Fork Trinity River, Hobo Gulch, Coffee Creek Road, Big Flat Meadow, Trinity River at Big French Creek, Scott Ranch, 3 miles from Trinity Center. Again, these are all places that for somebody who is from there, they might be, you know where they are, but we don't. And so we need your help. And so what I've asked Michael to do is to provide for you at the end of a survey, if you would like to be trained on how to georeference. Humboldt State is tasked with georeferencing the specimens from Northern California, and we can train you how to do that. We can give you access to their database, and you can put points on a map for where these collections are. It's a very wonderful detective exercise. Okay, that was my brief aside. Now, the other thing that we can get from herbarium specimens is an understanding of phenology. Phenology is the study of life timing events, and we are particularly working right now on understanding flowering time. When you look at a specimen, you can see if it has flowers on it. Most of them are collected when they are in flower. And what that tells us is that for that given species, at that day, at that place, that species was in flower. And that is incredibly important because many organisms, including humans, depend on plants for their resources, both flower, nectar, pollen, and fruits. And so this has led to the development of a project, a collaboration across California. It's called the California Phenology Project, or Capturing California's Flowers. And what we are trying to do is use digital images of herbarium specimens to study flowering time shifts in California and the Floristic Province. And so 2 years ago, we received a grant from the National Science Foundation. It's a 4-year-long project with the goal of building this historical record of flowering time using specimens that go back to the 1850s. What we're trying to understand with those specimens is which specimens might be in danger of a phenological mismatch. This is when the plant and the pollinator don't line up. Which habitats and vegetation types are most phenologically sensitive to climate change, and where might mismatches occur between plants and their pollinators, their pathogens, and their pests. And so what we are trying to do with these specimens is first we take high resolution images of them. We need help transcribing the label data. We georeference them, and then we say whether or not that specimen is in flower. And our goal is to do all of these things on 1 million herbarium specimens throughout California. So for the last 2 years, all of these institutions and a few more have been taking photographs of herbarium specimens, and we are very near our goal, actually. I'd like to plug the fact that in Northern California, you have a lot of institutions near you. So the institutions here in green are currently contributing their data to our database, and you can find the one closest to you, and that's where you should start to submit your samples, or you should go there to volunteer. It's especially a wonderful activity to do in the dead of winter when you need a little taste of spring. Most active in your region are Humboldt State with Mike Messler and Robin Benice. They are also tasked with georeferencing a third of these million specimens. And so they particularly want your help. Also CSU Chico with Colleen Hatfield and Lawrence Janeway, and the Mendocino Coast College Center with Teresa Scholars. Part of this project has been building a new database for California's Herbaria to share their digital data. And that new database, we're calling it the cch2.org to distinguish it from the CCH1 that we all know and love. And this is where institutions are putting these images, and they're now all searchable online. So you can go on and you can see specimens from every institution in California. It's an excellent shelter-at-home activity. So to lead our team in these endeavors, 2 very important people. Katie Pearson, who you heard a little bit about earlier, is tasked with coordinating all of these efforts. We've tried to leverage Katie's expertise and time to help every institution in California, whether you are part of our project or not. And so she does a lot of training and database management help to anyone. And if that applies to anybody who's listening, please be in touch with us. And then Jason Alexander is the database manager. He's based at UC Berkeley, and he is responsible for making sure that all of these infrastructural tools that everyone knows and love, like the JEPS and eFlora, CCH1 and CCH2, all are online and available and working for everybody. So we have this target of creating 1 million specimen records to understand flowering time shifts in California. And that brings me to the main point, which is that we need your help. And so at this point in the webinar, I'm going to switch over to Katie. And if you have the ability to both watch a webinar and manipulate a web browser, you can follow along with Katie as she does a little workshop into what you can do from your own home right now to help these efforts move forward. And with that, I will hand it over to Katie.
Thanks, Jen. Well, I'm going to share a very familiar slide. Here we are. So as Jen was saying, our goal, at least for this project, is 1 million specimens, but there are more than 1 million out there that need to be digitized, and we really need your help. So right now I'm going to talk about the transcription steps. So there are multiple aspects of digitization, but this is the taking the data from the label and putting it into the database fields. And while this may look a little complicated, don't worry, it's not. So once these data are in our database, they can be searchable by image, by their species, by their collection date, by whoever collected them. And then that's when the research, the education, the outreach, and just the everyday use can happen. So I'm going to tell you how to get involved through a very easy to use website called notesfromnature.org. So I'll have this URL throughout the talks, but you can look at it and type that into your web browser right now, try it out. And what Notes from Nature is it's a citizen science platform where multiple herbaria have their collections, have a project here. So we are one of many projects. This is us over here with the poppy. And within our project, we have multiple expeditions. And an expedition is a collection of specimens, usually from one or two specific institutions that are hoping to get your help in transcribing data. So these are the two expeditions we have going on right now. And if you clicked one of those buttons, and I'll show you that in a little bit, it'll come up with a page that'll look something like this, much more simple. All you have to do is take the information from this label and transcribe it into some fields that we ask for specifically. Once we have, or for every specimen, it is transcribed by 3 separate people. Because this is so open access, anyone with a web browser can transcribe these specimen labels. All 3 of those are kept, and then we use a consolidation algorithm to keep the best version of all 3 of those together. I do some data cleaning, and then I import them directly into CCH2, which Jen was talking about earlier. That's the live place where the data will live and be accessed for research and education. This project has so far been very successful. We've completed 6 expeditions, and 2 expeditions are ongoing right now with more on the way. We have transcribed over 10,000 specimen records. And remember, each specimen has to be transcribed 3 times. So that's over 31,000 specimen transcription events. We've engaged over 1,000 volunteers in our project alone from across even the globe. And this is a little bit of a silver lining actually for these COVID times. This arrow right here is the timing of the California shelter at home order. Because this is something you can do from your browser, you know, even while you're watching Netflix, we've seen a big bump in people getting on and entering data, looking at these specimens, even after this shelter at home order. So again, notesfromnature.org, click the poppy, look at one of our expeditions. The expeditions we have right now are from UC Irvine and Cal State LA. These are some really neat collections that have been previously digitized only a very tiny bit. So we have Rebecca Crow, who is also the manager of the UC Irvine Arboretum. If you've ever been down to the Irvine Arboretum, it's gorgeous. And the CNPS Orange County have been involved with digitization at UC Irvine as well. So really neat collection that has only been digitized probably a few %, but there are many, many thousands of images that are just waiting to be entered. And then at Cal State LA, this is our PI, Kirsten Fisher, and then the master's student, Joseph Zyla, who has been instrumental in making this project go forward at Cal State LA. And Cal State LA is, I wouldn't say unique, but it is one of the collections that had 0 digitization at the beginning of this grant. So everything you're seeing has just started as of 2018. Really neat to see thousands of images on the portal now and thousands of images being transcribed from people like you. So if you select one of these expeditions, what you'll see is something like this. This is the page that comes up. There's a tutorial in the very beginning right here. You can click through that. It'll give you much more information about exactly what we're looking for. And there's help along the way, the whole way. So there's this field guide over here on the far right. There's deeper explanations if you find some weird something that you have a question on. There's also for every field, there's a need help with this task help box. It'll give you an example of exactly what we're looking for and a little bit more information. So at this time, I'm going to open a browser and show it to you real time. And here we are. So here is notesfromnature.org. If I was coming on here for the very first time and I'm like, yeah, this sounds like something I really want to do, I would recommend registering. If you register, you're not going to get a bunch of spam. It's just so that you can keep track of your numbers. I have an embarrassingly large amount of notes from nature records. I don't know if it's embarrassing. I should be proud of that. Of records that I've transcribed over the years. And it's really neat to see those pile up and to see how many institutions I've been able to help through my work. Then once you've registered, you can sign in, have those numbers ticking up as you work. So I'm gonna scroll down. You can see there are lots of institutions with projects, but remember to go to the poppy. And then it'll pop up and you'll see this page. You can learn more about the project. You can explore the project. You can even look at statistics about how far we have gotten on these expeditions so far. You can look at how far we've come over time. That's what I was showing you earlier. But if you want to get started right away, you just got to go to this Get Started and select one of the expeditions. So I'm going to show you the kinds of things that we're asking for when you look at your, when you're typing these in. So here's the tutorial like I showed you. You can read through that at your leisure. And then here I have a specimen record and I have the fields that I want to enter. Okay, pro tip, do not just leave this right here, scroll all the way down, memorize lupinous bicolor and start typing it in. You can move this around. So scroll, 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 use your tools. This is gonna let you zoom in and out. Get it right up there, right next to what you want to enter, and then start entering away. So there are several panels that it's going to ask you for data for. So for this example, first it's going to ask me about the collector. If I was unsure, well, is the collector, where's the name usually? Of course, you can click need some help with this task, and there you go. So I can enter the collector number here. It's pretty obvious here. It says collected by. Then the collector number. I've gotten a challenging one and there's not a clear collector number on this. Well, in that case, I can leave it blank. Now I just need to enter the date of collection, and you don't have to select from the drop-down list. You can start typing and it will populate for you. There's my April 25th. 1958. There we go. And I can click next, and I can keep moving on. Then it's going to ask me things about the scientific name. And again, if you need some help, open it up. This is telling us that we don't need the authorship, which is the information that comes right after the scientific name usually. So here I have my genus, specific epithet. This is the authorship for the species. This is authorship for the infraspecific epithet, which is this Piper Smithii. So I enter only the scientific name part. Then it's going to ask me what if I see any annotations. And an annotation is someone else has come to that specimen and identified it and either said, yeah, it's definitely this species, or they have identified it as something else. And usually that will appear as a label that's on top of, on the specimen. Sometimes it's actually just writing. So I'll show you an example of one right now. This is what an annotation would look like. Here's the main specimen label. Someone else came in and identified it as this specific variety. Someone else came and re-identified it once the name changed. So I don't see any annotations here. I'm going to say no, and then it asks whether it's cultivated. That would be if it's in a garden or if it was planted, or if it's very clear that someone cultivated this somewhere. Generally, most times it's no. This just says near SEMA. It doesn't indicate anything. And then you just keep on keeping on. Again, you can type in information. You can use the tab button. That's what I'm pressing to go in between my fields. And moving my arrow keys up and down, I can put location information. If there's any altitude information or elevation, put that in there with the units. And then it asks whether there are coordinates, so like latitude or longitude, usually in decimal degrees or degrees, minutes, seconds. I don't see any of that here, so I'm going to say no. And then it asks some questions about whether there's habitat information, a plant description by the collector. You don't have to describe anything you see there. And any other notes, like if it's common or frequent, or if it was collected for a specific project or something like that. I don't see any additional notes, so I got it off easy this time. But there is a little bit of challenge, and this is one of the reasons why humans have to do this rather than just computers. Sometimes you'll have specimen labels in which it's very unclear the differentiation between the location and the habitat information. So habitat information is the environment in which the plant was found. So this could be, it's forested, it's riparian, it's a woodland, there are these species around it, and then a location would be more like 3 miles north of something, something, something. However, sometimes that information can go in both fields because you have, if you were trying to place a dot on the map, it would help to know that it's on a lake shore. So you would put in the location, okay, it's on this lake shore. And then you could also put that information in habitat because it is useful to the habitat for it to be lakeshore there. So as you go through, you're likely going to find 1,000,001 different ways. So here's a map on this one. Here is some, let me zoom in so you guys can see what I'm looking at. Here's a map. This one was collected in Arizona. This one has a determiner, which is a person who identified the plant who isn't necessarily the collector. And it also has township range section on it, which is an old survey system that has been used to designate the point of the collection, or at any point really. So there's tons of information, as Jen was talking about, on this label, making it rich with data. And we really want to be able to mine all these data to use them for research and education. And then of course, information about the flowering time and fruiting time, that will be done in another instance, but that's what we hopefully will use in the future once we have all this information about what species we're looking at and where these have been collected.
Katie,
so
will
that's
you show people how to get to the expeditions from the homepage again?
Here we are on Notes from Nature. This is just the homepage. Scroll down and you'll see all of the projects, and we are the poppy. And then once you're on our splash page, it's right under Get Started. There are these buttons here. They will change as expeditions change, but you'll always see these buttons. Click one of those and you can get started transcribing. All right, are there any questions?
So Katie and or Jen, there is a question here for you. By the way, the two of you, awesome presentation. Thank you for that. That was really great. If we could hear all those people clapping, it would be awesome. They're out there clapping in their rooms. The question is about optical character recognition, and has there been any efforts with that? What is the problem? So can you just talk a little bit about that? Because I know you guys have thought about that quite a bit.
Yeah, that is a fantastic question. And we do have the ability to use optical character recognition. We have that ability in our portal. The problem is, like I was showing you a little bit earlier, it's actually fairly difficult for a computer to parse different types of data into the types of fields that we're looking at. So if I show you this example once more, you can see that it says on gravelly flats and slopes, alpine fell fields, North Spur. This is information that's highly heterogeneous. and it's difficult for even a machine learning algorithm to parse it into the different fields that we are trying to parse it into.
Nice. Jen, do you have anything else to add with that, or I can go on to another question?
No, but I did see a question which I wanted to address, which was about SciNet. So if anybody has used SciNet, SciNet is a data portal in the of herbarium records for the whole Southwest. CCH2 is just a California version of SciNet. So they're based on the same platform. They have sort of the same exact functionality. And our California portal, CCH2, now talks to SciNet. And so you can actually search records from within either of those portals, or at least it will be that way very soon into the future. Go ahead, Matt.
Well, okay, so there's a question here from Dean Taylor, which is this I think you could probably field, Jen, which is a little bit of pushback on how the species in herbaria imaging was formulated or prioritized. Why did we not emphasize the 2394 CNPS rare plants?
Well, great question, Dean. Our goal is to get everything imaged. That's our secret goal. But to make a competitive grant application, one thing that we were concerned with is that the grant that California Herbaria got back in 2005 focused on those rare plants. And we were trying to, for this grant call, we had to emphasize new records that had never been digitized before. And since the rare species were the target of that first grant, we couldn't make a strong case for why we also needed to get those plants. But I do hope that we will get those plants, and many of them we will get in the process of doing our target taxa. But I should also say that most institutions who are imaging, we found it too onerous to pull out specific taxa. And so most of the small collections, and by small, I mean 100,000 records or less, are just going through their collection and imaging everything with no regard whatsoever to the target taxa on our project. It's only in the larger institutions that they have to pull out the specific target taxa just because they're resource limited. They couldn't possibly do their whole collections. And that's mostly at Cal Academy, Jepson, and Rancho Santa Ana.
Okay, another question here, which is, can volunteers get involved with transcribing the phenology?
We currently don't have any expeditions for phenology. And the reason for that is because we were able to develop a tool that mines phenological data from existing text descriptions. So when you're transcribing data and you put something that says flowers white because it's on the label, and you transcribe flowers white into the specimen, into that field, you're actually helping us with the phenology too.
Okay.
Oh, I'll just add to that, which is that Katie and I right now are teaching a class in which we are taking a group of undergraduates. Each of them picked a species, and they went into CCH2 and they tried to georeference those records, and they scored the phenology on those records by looking at the specimens themselves and saying whether they were in flower or fruit or just in bud. And then they actually tried to use those data for understanding how these plants are responding to various climatic variables. And there are still huge data gaps, so we do need help. I think most of our sample sizes were too small, and that largely comes from actually not having good georeferences on specimens. So it is very likely that in the future we will have the need to have people look at specimens and say whether they are in flower.
Nice. I'll butt in here and just say, then I'll ask you the question that it has not been typed, but I'm curious about it, is how's all this data, or is all this data being used in research?
Yeah, well, the class that Katie and I are using is really the first attempt to use this brand new data for research. One problem that we've seen over time is that individual researchers who care about some aspect of how climate change will affect plants will download all of the herbarium records and then score phenology in their own way. And then that sits as a file on their home computer, and it's not accessible by anybody else. And so really our goal with this project and the CCH2 portal is to keep the specimen record data with the specimens so that anybody can come back over time and re-download those data and take their own look at them. And so we're right at the beginning of that. We're in year 2. We hope to have our 1 million records 2 years from now when the project is over. And so I hope that the research capabilities just increase over time.
Nice, thank you, Jen. Mark Johnson makes a great point here. He says that many botanists do not collect or voucher or go out and collect things that are very common or weedy. It's not worth my time. It's everywhere kind of impression that people have when they're out on botanical expeditions or collecting plants. Jen mentioned, for instance, the limited collections for French broom in Northern California. Do you two have suggestions or recommendations of how we might change botanists' perception of collecting non-native weeds? And it's interesting on iNaturalist as well, you see the same thing. The iNaturalist has a very huge push towards rarity in the amount of collections that are made. Do you have suggestions for people?
Yeah, I would say one of the best parts about having a SciNet-like portal for California is the CCH2. What that allows these collections to do is access data from their home computer. And so you don't need to physically go into the herbarium to access the database associated with that herbarium. This is a long answer, but I'm coming around to it. So if you are out there as a botanist and you can or want to make plant collections, then you should associate yourself with those institutions. And maybe Katie can drop the website for Humboldt and Chico into the chat, or just reach out to us and we'll put you in touch with the PIs there. But what you can do then is within their data portal, you can actually enter your own collection data into their database from home, which has always been a huge barrier to this. So you can be making collections, you can be entering the data, and then you just drop those collections off and tell the curator, like, all my data is in your portal, you can print a label right now. And so it overcomes that barrier of like, you wrote it down on some random piece of paper and now you need to give that away. All of the data can be entered right away. And that makes it easier for people to get those records into Herbaria. And we absolutely have to be documenting non-native species. I mean, nobody likes to do it. And so, because we're all enamored with the Calicortus and the Clintonia. And so I don't know how to really change perceptions other than I'm trying to lower the barriers to entry for people just making plant collections in general.
Which is a nice segue to a question that Katie just typed the answer to, but you might as well say to everybody, as somebody asked about, how do I get into plant collecting? How do I go out and start that process? How do I learn how to make herbarium specimens and prepare herbarium specimens? You teach that all the time, Jen, and Katie, you teach people that. So why don't you two say something about that to everybody?
Well, one thing I'll say is that if you want to get started with plant collecting weeds is the easiest thing to do because you don't actually have to worry about any permits. And so that's the safest thing to do. I recently identified the weed growing in the cracks of my driveway. And then I looked in the CCH2 and there's only 7 collections in the entire state of this ubiquitous little plant. And I thought, oh my gosh, I need to start making a flora of the weeds of my house. That's very important, like even just your garden weeds. And so that's probably the easiest thing to do. All you need in order to make a plant collection is the ability to dry and flatten a plant quickly. And so we often just put a plant in newspaper and then we try to get that, we put it between cardboard and then you leave it in the backseat of your car under some weight. That's the simplest way that you can do it. And then what you'll accumulate is a bunch of plant specimens in newspaper. If you have the data associated with each of those, they're a valuable collection for any institution.
Great. Thank you. Question here. What are some BMPs you could touch on for submission to Herbaria during the COVID chronicles?
Well, all the herbaria are closed right now and nobody's allowed to go in them for the most part. That's probably true California wide at this point. And so I would say for the time being, hold on to your specimens. And as soon as things open up, you can drop them in a labeled box outside the building and flee.
It's a question here about using photos instead of specimens with all these data.
Yeah, absolutely. And so iNaturalist is probably the biggest platform where that takes place. And Matt and I, since we are right now teaching field botany, but online, are making heavy use of iNaturalist. And there are excellent scripts for downloading the iNaturalist data and really great, actually, computer learning algorithms that can score whether or not that image is of a flower or not. And so you can produce a ton of records about flowering time from iNaturalist. The only issue with iNaturalist is really that they are only recent observations. And so what we're trying to do with the herbarium specimens is build the historical record that we can then compare against today's records, which iNaturalist is very good for. The other thing that we've done is we've partnered with the National Phenology Network, and that's a group of citizens who adopt a plant near them of a given species list, and they record the phenology on that living plant in the ground over years, and they have a whole data sheet they fill out. Our target taxa are all of their taxa, and so that we can build the historical record for the in situ modern day plants, same as iNaturalist.
All right. Well, Katie Pearson, Jenn Yost, you guys are awesome. Thank you so much for that presentation and for your excitement about this topic and giving people some skills tonight as well as information. So
we expect to see a really big spike in the transcriptions, all coming from Northern California. After
represent
111 attendees, each of them is going to do 100 of these things, so that's a lot. So thank you all for being here. Thank you to Katie and Jenn and Michael and everybody from the chapter for having us on the call. So thank you all.
Thanks so much for having us.
Yep. Goodbye, you guys.